Hello guys, welcome to the channel. Today we will see what do you mean by out of specification or OOS. This is the most commonly asked question in the pharma company. So we will understand what does this mean. What do you mean by out of specification or OOS? So OOS means out of specification as the full form. So means that the test results for your sample, drug substances or drug product do not meet the accepted established criteria. So say for example we have any sample and we are going to test that, and we have a specification like percentage SA, percentage RSD, percentage purity, or other test whatever it is listed. It has certain criteria like 98 to 102, like 5% or 99.5%. If it doesn't fall within the acceptance criteria or this specification criteria for a particular sample for a particular test, then it is called out of specification. It is called OAS, right? So these criteria may be set by the either an official compendia like USP, FDA, ICH, or any other like JP, Indian Pharmacopoeia, any official compendia, or by your organization like Novartis, Dr. Reddy or Vindo, wherever you are working, or by the testing laboratory, whichever the laboratory you are working, individual laboratory. So next, can you explain OAS with an example? This is very commonly asked question. So for example, the specification limit or acceptance criteria for assay test parameter of a particular batch is this much, 98 to 102%, say suppose assay criteria, right? But the test results obtained, but the results what any analyst is obtained is 97.2. Then this result is not meeting the acceptance criteria and is called the out of specification. As I have already told, if it is not within this acceptance criteria, then it is out of specification and it is called OOS, right? Then if you see another example, tolmetin sodium for the organic impurities test parameter, say suppose for the acceptance criteria is 5% and replicates for not more than 5%, and the result we have obtained is 6.78%, right? Then it is failing our criteria, then it is OOS. What could be the reason of OOS results? We have seen an example, but what could be the actual reason of OOS results? So here some issues with the operating instrument, whatever the instrument we are using like HPLC, UV, IR, GC, the operating condition of the system is not good, or it could be the issue with the procedure of the product tested. So procedure whatever we are testing like tolmetin sodium assay, the diluent is not good, the solvent is not good, or column is not good, or the peak shape of the sample is not good, the procedure is not good, right? It could be issue with the sample like contamination or sample homogeneity. So if our sample is contaminated, whichever the sample we are testing it is contaminated, it will not give resolution, or it will give some splitted peaks due to impure, or a sample is not homogeneously mixed, then it will also give the issue with the analyst or scientist. Whenever the scientist is working, he has not done proper dilution or proper calculation or proper pipetting or proper transfer of vials, so that could be the reason of the OAS. What is the purpose of OAS investigation? So we have seen few other things. This is also important. The main purpose of OAS investigation is to identify the root cause of the OAS results and to take appropriate action like corrective action, preventive action. This I will describe in some other videos. The investigation should include following reviews: error related to the analytical laboratory, right? This should describe this also. Analytical laboratory, the validated procedure was not followed correctly, or deviation from the standard procedure, right? So if the procedure said take 1 ml and dilute it to 10 ml, but you have done 2 ml and dilute it to 10 ml, so this is a procedure deviation, right? Analyst error, same, it will cover into it. The analyst has done any error while doing calculations, we have not done proper calculations, then also it will be an OAS investigation. Error due to the instrument failure, suppose we have HPLC and it is continuously running and in between it got failed, or the power got tripped, so this could be the instrument failure error. The test sample is not uniform or homogeneous, as I have discussed already. Quality of the used excipients, raw materials and intermediates is not good, like we have to go from A to B to C to D, so the raw materials which are used in this is not good, this is not pure, this is only 80% pure or 90% pure, but we are doing calculation according to the 100%, so it can also cause to the failure and it could lead to the OAS investigation. What are the investigation phases of OAS as per USFDA? So USFDA has given different phases for the OAS investigation, so we need to understand this. because USFDA is a very important regulatory agency, right? So phase 1 investigation, laboratory investigation. So whatever the samples we have analyzed, right, so we have to investigate for these samples, like sample is correct or not, sample is expired or not, how much sample weight we have taken, how much dilution we have taken, how much volume we have maintained. So these things needs to be investigated in the laboratory. If here it is not cleared, if we are not getting any conclusion, then we'll move to the phase 2 investigation, full scale investigation. In full scale investigation there are two types, A and B. Manufacturing process review, whatever the before coming to the testing to the validation stage or some other stage, we have done manufacturing, right? Some manufacturing, some pilot scale it will be carried out, so we have to first see there also whatever the problem. So manufacturing process we have to review. All department involved in production, so suppose production is going on, so QA, QC, procurement and dispatch, whatever the departments are involved, this we have to investigate. And phase 2B, this is going to be a very large scope to be investigate. Additional laboratory testing, retesting and resampling. If we are not yet clear into this stage, then we have to do retest and resampling. This is going to be a very big issue for any pharmaceutical company. So if you like the video, please like and share with your friends, subscribe the channel for more content. Thank you for listening. Thank you all.